Early life exposure to the SSRI paroxetine exacerbates depression-like behavior in anxiety and depression-prone rats. According to the World Health Organization, over 300 million people around the world have depression. The mood disorder disproportionately affects women, with women being 70% more likely to develop depression than men. Among the women affected, 10 to 20% of pregnant women experience depression. One of the most prominent treatments for depression is medication, specifically selective serotonin reuptake inhibitors, or SSRIs. While these drugs can be effective for decreasing depression, they have also been linked to problems for children whose mothers take SSRIs during pregnancy. In 2005, the Food and Drug Administration issued a warning against using the SSRI paroxetine during pregnancy because of fetal cardiovascular abnormalities. There have been a multitude of reports of fetal problems linked to SSRI use, including neonatal withdrawal syndrome, increased risk of autism, social avoidance, and psychomotor defects. While direct explanation is not known, it is possible that SSRI interference with the 5-HT is causing the lifelong brain development problems. In past studies, only certain individuals develop depression after exposure to SSRIs during gestation. These studies are helpful to increase the base of knowledge, however, they are inconclusive as to why only some individuals were affected. In Clinton's study, a major goal was to model these differences to determine individual vulnerability. In order to simulate individual differences, the researchers used rats bred for low response to novelty and rats bred for high response to novelty. Low responder rats display anxiety and depression-like behavior, which is attributed to their lower levels of SIRT and TPH2 mRNA and increase of 5-HT receptors. The high responder rats do not display these behaviors. In this experiment, it was first hypothesized that treating the low-responding rats with paroxetine would improve their depression. It was also hypothesized that by exposing young, low-responding rats to SSRIs, that they would be at increased risk of depression. Experiment 1 focuses on the effect of paroxetine on the adult rats and analyzes how they were affected. The SSRI paroxetine was administered at a rate of 10 milligrams per kilogram per day, and tap water was used for control. The rats were given either the paroxetine or the control for a period of 30 days, after which they were evaluated using behavioral test battery. The rats were administered a forced swim test, which measures the level of depression-like behavior based on how a rat behaves in a container full of water. If a rat is immobile and exhibits no other effort than to keep them afloat, that is an indication of depression-like behavior. The rats were evaluated using the open field test. The open field test measures the behavior of a rat in an enclosed space for 5 minutes, recording how long they stay in each position. The locomotor response to novelty for the rats was recorded from this procedure. The elevated plus maze is a plexiglass black box containing 4 elevated arms. The rats' movement was tracked for 3.5 hours and measured the anxiety levels of the rats. After all of the behavioral tests, rat brains were collected and analyzed to view levels of paroxetine. In the second experiment, the effects of the SSRI paroxetine during pregnancy on the behavior of offspring was tested. Adult low-responding and high-responding rats were administered paroxetine at a rate of 10 milligrams per kilogram per day, both before pregnancy, during pregnancy, and during feeding of the pups. The pups were weaned 21 days after birth. Once the pups reached adulthood, they underwent behavioral test battery using the same procedures as during the preliminary experiment. In the third experiment, rat pups were bred using the same procedures as in the previous parts. However, in this experiment, no behavioral tests were done. Rat brains were collected at several different critical development points: 7 days after birth, 14 days after birth, 21 days after birth, and 75 days after birth. Brains were frozen and sliced using a cryostat and crescent violet stained. Samples from the hippocampus and amygdala regions were taken, and RNA was extracted. cDNA was processed and analyzed through NimbleScan, and genetic expression data was determined. During the first experiment, it was found that paroxetine worked for reducing the depression of the low-responding rats. The drug had no effect on the high-responding rats, likely because they did not have any depression-like symptoms to be treated. In the figure shown, it is clear that the low responders had overall higher depression and anxiety levels than the high responders, both before and after treatment. Figures B, C, D, and E diagram the results from each behavior test, though the paroxetine did not affect the performance for all the tests. The most notable test was the forced swim test, demonstrated by figure B, which shows a statistically significant difference between the performance of the low responders with and without treatment. When the rats were treated, they spent less of their time in the water immobile, which points to lower depression-like behavior. Though not specifically outlined as part of the experiment, the effect of paroxetine on maternal behavior was monitored. Maternal behavior can affect the future behavior and emotional state of the pup, so it is important to verify that it was not affecting the depression of the pups. For the most part, maternal behavior was consistent. However, for paroxetine-treated low-responding mothers, nursing was significantly less passive. In experiment 2, it was found that exposure to paroxetine during gestation will increase the depression-like behavior of low-responding rats. The high-responding rats were not affected. Figure B shows the results of the forced swim test for pups with and without perinatal paroxetine exposure, indicating a significant effect of the paroxetine. The low-responding rats spent about 20% more time immobile in the water when exposed to paroxetine during pregnancy, a clear increase in the depression-like behavior of the rats. In experiment 3, rat brains were extracted and analyzed in order to view gene expression. There were significant gene expression changes in rats who were exposed to paroxetine and those that were not. Figure C shows that the amygdala gene expression was affected, with the most changes found at postnatal day 21, in which about 200 genes were different. Paroxetine had a much greater effect on the hippocampal gene expression, with about 700 genes being affected after the 7th day after birth, though the differences did decline as the pup grew. Figure D shows that most of the changes in gene expression were down-regulated in the amygdala, while most were down-regulated in the hippocampus until P75, in which the majority switched to up-regulated genes. The differences in gene expression were determined to be biologically significant. In order to further examine the differences in gene expression, Venn diagrams were created to compare genes altered through development in both the amygdala and hippocampus. Gene clusters in the amygdala and hippocampus were created and analyzed to identify functionally related genes. The exposure to paroxetine during pregnancy can cause significant lifelong behavioral effects for offspring. It can cause a multitude of differences in gene expression, causing problems in mitochondrial function, synaptic vesicle transport and release, and neurogenesis. The hippocampal genes tend to be more affected by the paroxetine than the amygdala, likely due to the differences in density of the 5-H-tergic fibers in these locations. In previous studies, individual differences were noted, but in this study, they were further explored using high-responder rats and low-responder rats. The low-responder rats were greatly affected by the paroxetine, while the high-responders were not. This can be related to how some human babies are affected, while others are not. It depends on the predisposition of the individual. While paroxetine was the SSRI explored in this study, there are a number of antidepressants, which are each likely to affect offspring behavior in slightly different ways. In this study, the researchers noted some of the limitations that they found. Because of the low-responding rats' predisposition to anxiety and depression, it is possible that there is a ceiling effect, and that the data exploring the rats' exposure to paroxetine was slightly skewed. The researchers also noted some further research that should be explored in order to gain a wealth of knowledge about SSRI effects on development. It would be important to test brain development and behavior responding to different types of SSRIs to see the effects and how they vary based on the medication. Furthermore, most research done in this area focuses on the male offspring and not females. First, it is important to equally test both sexes in order to gain a complete data set. And furthermore, females are much more likely to be affected by depression and anxiety than males. The results for female SSRI exposure may be dramatically different than males, and could provide some insight as to why females have a greater risk of depression. There are several improvements that the researchers could have made or could execute in further studies. While they did mention their limitation of not using female pups, the importance of this cannot be overstated. Females need to be tested just as frequently as males, if not more, because of their heightened risk of depression. Furthermore, it would be interesting to see if the dosage of SSRI needed for males or females would need to be adjusted in order to have the same effect on depression. One problem with this study is that while the researchers used 4 types of tests to analyze depression and anxiety-like behavior, only one of the tests produced any significant results. The other tests seemed to not be affected by the addition of paroxetine. In order to gain a full picture of the effects of paroxetine, more tests should be utilized, specifically one that focuses on anxiety. The anxiety test in the experiment was not affected by the addition of paroxetine at all.